So I'm Frederic Chereau, I'm the CEO of LogicBio, and we define ourselves as a genome editing technology company, a little bit by opposition to gene therapy and gene editing with the CRISPRs and other nucleases you may have heard about. And we believe we have developed a technology which is very safe, and that's the reason why we have decided to target pediatric diseases. And as you know, all of these diseases are acquired, so it's important to intervene very early on in the life of the patients. So we became a gene therapy we became a public company in October last year, so here is our disclaimer. So our platform technology is called GeneRide, and it's a genome editing proprietary platform. And as you will see, with one injection, one vector, and most of the time in one-day-old mice, we can provide lifelong protein expression. The lead program is for methylmalonic acidemia. It's a rare organic acidemia, and I will tell you more about that. But it's really a platform we have, as you will see, where you can, it's broadly applicable in the liver, in other tissues, and to target many kind of diseases. We have assembled a team of 34 people now. We are based in Cambridge, Mass, as you can tell with my accent, I guess. And we have been founded originally by OrbiMed and became public, as I said, last year. So GeneRide is a platform which is nuclease-free, which means we don't need any kind of Cas9 or zinc finger or nucleases. And we are leveraging a naturally occurring process called homologous recombination. This technique allows us to have a very site-specific integration, so very safe, as I was saying before. And in addition of not using nucleases, we don't need to carry with us, or with our technology, any exogenous promoter. We take advantage of some endogenous promoter which are already in the cells when we intervene. So here it's the way it works. On the right side, on the top is our product. In the middle, in orange, is the gene. In this case, it's MUT for methylmalonic acidemia. It's flanked by a small 2-peptide called 2A peptide. It's 20 base pair. And then it's surrounded by 2 homology arms. These homology arms are about 500 to 1,500 nucleotides. And we build them in a way to mimic or to mirror the sequence of DNA we want to integrate into the genome. In the case of the liver, we have decided to intervene at the very end of the albumin locus, just before the stop codon, so we build these arms in that way. So by homologous recombination, where our technology comes in front of this part of the albumin locus, it does integrate in a non-disruptive manner. So if you look at the top line here, you have the full DNA with the albumin gene, the 2A, and the MUT, just before the stop codon. On an mRNA standpoint, it gives you a long mRNA with the albumin, the 2A, and the MUT again. And the MUT plays a role as the protein expression. It does what is called a bicistronic expression. With one, it's doing ribosomal skipping. Basically, it's when the ribosome starts to read the albumin and then the 2A, at the end of the albumin, at the end of the 2A, it's a self-folding peptide, and the ribosome can continue its work and express the protein of choice with a 1-to-1 ratio, and it's almost error-free. At least we have never detected any errors in our experiments. So a very simple way to integrate and then to express the protein expression. So some could claim that homologous recombination is not very efficient as a process, and that's true. People agree to say it's about 0.1 to 1% of the cells which can be modified by homologous recombination. So how can we express enough protein with a limited number of cells being modified in the liver? That's where, that's the second part of our technology. We have decided to take a ride on a very potent gene, and the albumin in the case of the liver, other genes in the case of other tissues. And as you know, albumin is the most expressed gene in the liver. You can see on this graph that among the top 2,000 genes expressed in the liver, albumin is at the very top. And about 90–95% of all the other genes are below 1% of albumin. So what does that mean? That means in terms of protein expression, if you compare albumin to other genes, PAH for PKU, factor IX for hemophilia, or MUT for MMA, it's dwarfed completely in terms of expression what you need to express for these diseases. And you can see a small line in orange, which represents 1% of albumin. And if I focus on this line, you can see that 1% of albumin is as much as physiological level of PAH, factor IX, and way more than MUT or UGT1A1, which is for Crigler-Najjar. So small integration, but a big engine behind it, and protein expression is just the product of the integration times the expression of the gene you are using, and here, once again, albumin in the case of the liver. So I was talking about the modularity of the platform, and you have two main pieces, the capsid on the right-hand side and the cassette. So we will focus first on the cassette, and I already described how the cassette is made. But when you want to move from one disease to another, the only thing you have to do is to swap out the orange part, the transgene, and swap in a new one. And we have demonstrated that in many diseases, as you can see here, and more, which has been published, and more than that in our labs, which will be published hopefully soon. So very modular. At least when you decide to target the same tissue, you can use the same capsid and the same homology arms. If you decide to target another tissue, obviously you may need another capsid, but we are totally capsid agnostic, so you can pick whatever capsid is the best for what you try to achieve. And here we just have to change the homology arms. We build new homology arms in order to target another gene that we're going to take a ride on once again. And the capsid is the last part of our construct, and very important part. As you know, most of the capsid until recently have been developed or selected for their propensity to target mice tissues, because that was the only way we could do until recently. And we know that in the case of AAV8, for example, it's a great capsid to target the liver of the mice. It's not as good for NHP, non-human primate, and it's not as good for humans. So what the team at Stanford, where the technology is coming from, has done, they use this chimeric mice, which is a mice with a human liver. And then they use the library of thousands of capsid, which has been developed by the shuffling technique, and they selected the capsid which has been targeting in the most efficiently the human cells within the mice. And so we know that LKO3, which is the name of this capsid, is, at least in this model, by far the best capsid to target human cells. It is currently in the clinic. Actually, a company called Spark is using it in hemophilia A program, and I will refer to them, to their program and their data if you want to look at the efficacy. But there are a lot of data on this capsid, and it's great for us because we already took it through manufacturing, regulatory, and now clinical. So here is our pipeline. As I said, methylmalonic acidemia is our lead compound. We expect to file the IND by the end of this year. And we have a very powerful discovery engine process. In blue is everything which is related to the liver. As I said, you don't need to change the capsid, you don't need to change the homology arms, you just swap out and swap out the gene and swap in another one. We have a lot of ongoing programs for different models. If we decide to go to another tissue, such as muscle and CNS, we don't need a new capsid right now. We can use what is on the market, but we can do already some proof of concept. So these three indications have been already published, so that's the reason why they are here. And here it's hemophilia B for pediatric patient population, because as I think you know, one of the problems of gene therapy is the protein expression durability. And when you are inserting into the genome, you don't face this problem anymore. So that allows us to go very early on in patients. So a few words about methylmalonic acidemia. It's a terrible disease. The patients I mean, there is a mutation in a gene called MUT, as I was saying, and this mutation prevents them to digest or metabolize proteins, amino acids, branch amino acids, exactly. And so if you don't give them proteins, there is no growth. If you give them proteins, you trigger metabolic storms, which leads to coma, developmental issues, and death. These patients are living with formula all their life, sometimes administered through GI tubes, and ultimately the last thing they can go for is liver transplantation, which has, as they say, always an expiry date. So, I mean, great unmet need here, and the patient population is about 15,000 to 2,000 patients in the US, about 3,000 patients in the rest of the Western world. So very good disease for us because we can uniquely intervene very early in the life of these patients. There is newborn screening in the US, not in so many countries in Europe, but even without the newborn screening, the mothers realize that there is something going on with the kids within the first weeks. When are we going to be able to intervene is to be defined, and we have some more work to do. But the idea would be to intervene as soon as possible before some metabolic storms occurs and some developmental issues are non-reversible. Here are some data in our mice model. As you can see, at 1 month, we have a significant difference in weight and also in multiple, but here it's one of the metabolites, sorry. But more importantly, what is interesting is in this case, it's 12 months on these pictures. On the right-hand side, we see a great colonization or a higher rate of modified cells. Maybe I should go on the pictures in the middle. It is what you see after a few weeks. And I don't know if you can see on this slide, but there are about 1% of the cells in brown which has been modified. What we know with this disease and with others, we have a selective advantage, which means when the cells have been modified and when the liver is growing or regenerating, the modified one take over the non-modified one. They are in better health in some ways, and in this case, it's a mitochondrial health. And what we see after a few months, or 12 months in the case of this picture, we see that up to 24%, according to a pathologist, of the hepatocytes in the liver has been colonized. So we start with 1 or 0.1 to 1, and we end up and we plateau most of the time at 25%, which is plenty enough. We don't need it, but it's a good feature for some specific disease. We see that only in diseases where the hepatocytes are sick. We don't see that in diseases where you use the hepatocytes as a factory to produce the missing protein. Sorry. And the last slide is highlight. So as I said, I mean, we are very different than many other technologies you may have seen, and very powerful product engine. And today, the main focus is on MMA, but I think I've shared with you some other diseases we could be interested in. And many progress have been done over the past 3 years. The company is 3 years old, and we have a lot of development milestones for this year with the nomination of a second candidate for a second indication. We have the retrospective chart review for MMA, or natural history study, sorry. Potentially some strategic partnership, and by the end of the year, filing an IND in the US. Thank you.